Okay, so this year, this year's 2026 JAMB biology revision, we're going to look at this first question here. If you have this question before you, what do you do? Now let's read. Which of the following is an example of a carnivorous plant? A carnivorous plant. You have option A, Hydra, B, bladderwort, C, yeast, D, spirogyra. Now, what is a carnivorous plant? That's the first thing that will be going through your mind. Now, you need to know what it means, and then you need to know which of them is a carnivorous plant. From the word carnivores, they are flesh eaters, or they eat only anything that has flesh. That means they can't eat plants, only flesh. But you'll be asking yourself, what kind of dentition does a plant have to eat flesh or anything that has flesh? Alright, they don't actually have a dentition like the powerful premolar and molar or a canine to tear flesh, but they have special devices. Yeah, they do. Special devices they can use to trap insects. First of all, to lure the insects, attract them. When they attract them, then they can feed on them. So we have some plants. Plants like Venus flytrap. Yes, here in the picture is a Venus flytrap. It has special devices. You can see it's actually brightly colored to attract insects like flies and the rest. And when they perch on it, it's sensitive to touch. Immediately it closes, and then it digests the insects by secreting some enzymes on it, more like what we call extracellular digestion, and then it can feed on that insect. We also have another one we call the utricularia, also called bladderworts. Yes. So a bladderwort, as we see in option B, is a plant that has a special device like a kind of it can secrete a kind of air bubble or a kind of sac-like bubble that it's in water area or aquatic area. It could trap microscopic algae and feed on them. Then we also have, there are so many other ones. But if you look at this option, another way you can answer is that you need to spot out the ones that are plants, because the question says a carnivorous plant. So the ones that are plants are actually, Hydra is not a plant. Yes, Hydra is what we call an invertebrate. Yes, let me go closer so that I can write that. A Hydra is an invertebrate. So what does it mean? That a hydra is an organism that does not have a backbone per se. Invertebrates do not have backbones. They are kind of smaller organisms that they can move to everything. They're mostly animals, but they don't have a backbone. So a hydra is one. Hydra has a kind of tentacle. It can feed opening here, more like an anal opening or a mouth opening, something like that. And then hydra can feed in here, but it's not a plant, so not a plant. And remember, the question says here a carnivorous plant, so plant is the keyword. So of course hydra is off. Another one is yeast. Yeast is not a plant. What is yeast? Yeast is a type of fungi. Yes, it's a type of fungi. And you know that fungi, they belong to their own kingdom. They are not under kingdom Plantae. You can also use yeast in baking and all that. Can also improve the eyesight. Then the bladderwort, yes, that's the one I said. We also call it utricularia. Utricularia or bladderworts can help to trap certain microscopic organisms in water using something like an air bubble, and then they trap them inside. Then a spirogyra is a plant, yes, but it is not a carnivorous plant. Now a spirogyra is mostly belongs to the division we call Talophyta, under the divisions of plants. Talophyta, they are microscopic. They have structures that look like a root, stem, and leaf, but they are not true plants. And they can also produce food, but they do not belong to carnivorous plants. So that is it. Let's go to the next question. All right, number 2 question, still on 2026 JAMB biology revision. The question here says, emulsification of fats takes place in the part labeled dash. Now, if you look at this diagram, you need to know where this diagram is coming from. Of course, because if you're not sure, for the fats, they've mentioned fats. This diagram is coming from a part of we use the digestive system or alimentary canal. But here's what I like to do. I like to use one question to cover as many topics that I can. Now, you would call this question coming from a part of the digestive system, because the digestive system is a large system under which the alimentary canal is involved or is part of. Because the actual difference is that difference between alimentary canal and digestive system is this. Alimentary canal are the pathways through which food passes from the beginning of where that food enters the mouth to the end when it's out of the anus after digestion, and the undigested part passes out of the body through the anus. So all those pathways are the alimentary canal. So anywhere food does not pass through, whether it's digested or undigested food, is not part of the alimentary canal. Unless the food passes there, then it's part of the alimentary canal. Do you understand? Then the digestive system are all parts that are involved in the digestion of that food, even if the food passes or it doesn't pass. Now, this structure right here is the liver. This one here, this large, it's actually the largest organ in the body, the liver. Why this one here is the stomach. So the stomach actually overlaps the liver. It kind of covers a part of the liver. Why here is coming from the esophagus, from the throat. So the food comes in from the mouth, pharynx, passes down through the larynx, pharynx, esophagus, gets to the stomach. From here, the pyloric splinter opens somewhere here, and then the food can move into the first part of the small intestine, which is called here the duodenum. This organ that looks like a leaf-like structure, this is the pancreas. It actually secretes three enzymes that are found in a fluid we call pancreatic juice. So those three enzymes actually digest carbohydrate, protein, and then fat and oil. But the question now is asking us not on just digestion of fats or fat and oil, but on emulsification of fats. So what is emulsification of fats? Emulsification means to break fats, because fats are actually large globules made up of fatty acids, made up of triglyceride bonds. So they need to be broken. So breaking it down into tiny droplets is what we call emulsification of fats, and it is always done with the help of a greenish substance produced by the liver we call the bile. So the bile, this is what I mean, bile, the bile. So this structure here, bile, is actually produced by this guy here, liver, and stored here, called the gallbladder. So whenever this liver the bile is stored here, it means for the fat it was even produced is actually needed. Sometimes this bile is also gotten from destruction of red blood cells, which is done by the liver and the spleen filters, and then most of this bile is stored there. So this first part of the small intestine, the small intestine has three parts. I used to tell students it's DG, ileum. So remember the three parts of the small intestine. This actually is the first part, called the duodenum, the jejunum, and the ileum. So this first part here is the duodenum. It is actually here that emulsification of fat occurs. And what I say is emulsification of fat, I said is the breaking down of fat into smaller droplets before it can now be broken down by this enzyme here that is produced by the pancreas. An enzyme, there are actually three enzymes, but one of them will act on the fat and oil here after it has been fat and oil here. One of these enzymes produced by the pancreas will act on the fat and oil here after it has been emulsified by the bile. So the bile stored in the gallbladder leaves the gallbladder through this structure we call the bile ducts. The bile ducts enables the bile move and leave the gallbladder and moves to the first part of the small intestine here called duodenum. So this is where emulsification of fat occurs, not where it is, not where the gallbladder, not where the bile is stored. It is not occurring in the gallbladder, but it occurs in the duodenum here. So the answer to this question is actually IV, which is option D. This is where emulsification of fat occurs. Do you understand? There are still more questions on this diagram. So because of the space I have on my board, I'm going to wipe this question off, draw and write out the next question, and then tackle it with you. Stay tuned. All right, this is another question still on 2026 JAMB biology revision. Question number 4, it says, which of the following describes the sequence of blood flow from the heart to its tissue? There are several options, A, B, C, D. But the first thing you need to know, this question is coming from where? From the circulatory system. If it's just in plants, we call it a transport system. But if in humans, we use the words or animals, circulatory system, the flow of blood from where it's pumped from the heart to all parts of the body. One thing we must know is this. Now, whenever the heart is taking blood, the heart pumps blood and it wants to leave the heart to other parts of the body, it is usually leaving the heart through a major artery. Are you following? So the artery they are used should be aorta, AORTA. It's a major artery, and the blood actually leaves the heart through the left part of the heart, the lower left chamber of the heart called the left ventricle. Whenever the blood leaves, it's leaving through the artery, major artery aorta, and then it enters the, what's it called? The smaller artery is called arterioles. I'm looking at option A. If you look at option A, you would see what I'm talking about. So option A, we have the heart, we have the major artery, which is aorta. When they say artery, it's just the option they give, but this is what it should be like. The aorta, the major artery, then it goes to the little arteries we call arterioles, then the tissue, but any tissue. But before it gets to the tissue, there's usually a capillary here. Because actually the capillary is made up of these two, tiny arteries and tiny veins. So this word venular venules are out. So this is when the blood is returning back, more like deoxygenated blood returning from the tissue. It has to go this way if it's going through the venular veins and then the heart. And then it's coming in, of course, through the into the right chamber of the heart, not the left. So these options are wrong. Option B is wrong. Option C is wrong because it's dealing with the vein. The only two options that could confuse you a bit is A and D because they're dealing with arteries. But because this guy is starting with the little arteries, you don't need to bother yourself with it. So the answer to this is option A automatically. So hint here is anytime it's leaving the heart, because the question says the sequence of blood flow from the heart, that means it's moving from the heart to the tissue. It must start with major artery, or if they don't give you the major artery called aorta, you can give you artery, but not these little ones called arterioles. From there it gets to capillaries. From capillary it now gets to the tissue. But if it's coming back, returning to the heart, then it will move through veins. But first of all, from the tissue to capillary, that's option B. Tissue to capillary, capillary toward tiny veins we call venule. Then from venule to vein, major veins, and then to the heart. And what part of the heart is going to go in through the right heart, the right chamber of the heart. So for this question, which of the following describes the sequence of blood flow from the heart to a tissue? The answer is option E, which is A, which is heart, artery, arterioles. There should be capillaries, but because of the option, and then tissue. Thank you. Okay, still on 2026 biology JAMB. Question number 5, it says, which of the following groups of cells is devoid of true nuclei? A, algae, B, monera, C, fungi, D, viruses. Now, for something to have a nucleus, a true nucleus at that, it must be a cell. Because we are taught, or we already know that what is a cell. It must be a living thing. Yes, it must be a living thing for something to have a nuclei, because the nuclei is part of the cell. Are you following? And anything that has a nuclei has a cell, and anything that has a cell is a living thing. After all, we're taught characteristics of living things is that they feed, grow, do all these things, and what actually feed, grow, die, excrete is actually the individual cell, if it is a unicellular organism, or different cells if it's a multicellular organism. Now, We'll start with algae. Algae, what are they? Algae are actually microscopic. They are plants because they have the chlorophyll, but they're not true plants. They do not have true root stems and leaves. They are also under the kingdom or division rather, kingdom Plantae and division Talophyta. Then the thing about them is that because they are actually living things, they have the nuclei. They are plants and they are living things. They have the nucleus or the nuclei, singular, plural. Then the next one, option B, Monera. What are Moneras? Moneras are bacteria. Bacteria are known to have the nuclei because they are living things. Although most of them they do not have a nucleus, or rather a cell that has all the organelle. They just have the ribosomes and few other the cytoplasm and few other organelles, but they don't have much organelles. Then the next one, option C, fungi. They're actually living things. They have their own kingdom, belong to kingdom fungi. For algae, they belong to kingdom Plantae. For Monera, they stand on their own kingdom Monera or kingdom bacteria. Then the fungi, like we're talking about, they are living things, so they have nuclei. Yes, they have cells. A group of cells we call that are in hair-like form we call, or thread-like form we call mycelia, or mycelium. Then the next one we have, we call them, the next option D is virus. Viruses are not actually living things per se. Actually, what do I mean? They can be living things. They can also be, they can also not be living things, depending on where they are found. If they are found in or on the body of a living thing, they're actually a living thing. So they start behaving like a living thing. They can feed, grow, replicate, more like reproduce. Their DNA or RNA, but they are not living things when they're found on the surfaces of table, chair, anything that is not a living thing. So whenever they're found in those areas, we call them a viron. A viron is spelled, so we call them a viron, and a viron is spelled viron, viron. So this is when they are on the body or surface of non-living things. So the answer to this question is actually true. Devoid, that is lacking true nuclei, is a virus. A virus is the answer. Let's go to the last question for today. Please do remember to drop in the comment section topics you want me to teach in preparation for the 2026 JAMB, and also review past questions from those topics after teaching. And then I would also do well to help you all through your JAMB and certainly during your WAEC. So we'll look at one more question and then we call it a day. And then keep reading, keep studying, and I'll keep praying for you. All right, so the last question for today, it says, question number 6 says, alkaline pyrogallo was used in an experiment that experiment must have been connected with dash: A, excretion; B, germination; C, transpiration; D, digestion; E, photosynthesis. Now, what is alkaline pyrogallo? You need to know that it's a chemical substance used to remove oxygen. Whenever you want to remove oxygen, it helps to kind of like absorb or remove oxygen. Now, which of these processes do you think you might want to show that oxygen is needed? For excretion, no, because it has to do with removal of wastes, undigested waste. For excretion, it has to do with removal of metabolic waste like sweat, urine. So it's not really necessary. For B, germination, it's yes, because why is that so? Remember that there are some conditions necessary for germination to occur. One of them is what? Temperature, moisture, water, oxygen. Yes, these are the things that are necessary and are needed. The next one we have is transpiration, that water is given off, a process by which water is given off in form of vapor through the stomatas and the leaves. So that's not really necessary that we need alkaline pyrogallo. The next one is digestion, not needed. Then photosynthesis, yeah, oxygen gas is given off as a byproduct, but you do not need to remove the oxygen. You can only say you just collect it and test the gas to be sure that it's oxygen gas. If you want to, know the gases that are, released. So the answer to this question is actually B, which is germination. Thank you so much for today's section with me. Do well to stay tuned to Callista's bio class and drop in the comment section any topic you feel that you don't understand and you want me to teach and then revise with you. Bye bye.